Medical authorities in the German city of Düsseldorf are currently responding to a high-priority public health situation, following reports that a patient with a suspected novel strain of hantavirus has been admitted to a specialized bio-isolation unit. While official details regarding the patient's identity and specific travel history remain restricted due to privacy protocols and the ongoing nature of the clinical investigation, the activation of high-level containment measures underscores the seriousness with which German health agencies approach viral pathogens of this nature. Hantaviruses represent a significant category of zoonotic diseases, meaning they are transmitted from animals, specifically rodents, to humans. In the European context, hantaviruses are typically associated with hemorrhagic fever with renal syndrome, or HFARS, though the severity of the illness can vary significantly depending on the specific viral strain involved. The mention of a potentially new or previously undocumented variant in the Düsseldorf case has alerted the broader scientific community, as the emergence of a new strain could imply changes in virulence, transmission dynamics, or geographic distribution. For viewers and the general public, the significance of this development lies in the potential for hantaviruses to cause severe respiratory or renal distress. Most known infections occur through the inhalation of aerosolized particles from the waste of infected rodents, making certain rural and suburban activities higher risk. In Germany, hantavirus is not an unknown entity. Regions such as Lower Bavaria, Western Thuringia, and parts of Baden-Württemberg have historically reported clusters of the Pumala virus, which is carried by the bank vole. However, a case necessitating immediate bio-isolation in a major urban centre like Düsseldorf's suggests either a presentation of unusually severe symptoms or a clinical suspicion that the pathogen does not match the profiles of common endemic strains. This framing of the event as a suspected new variant places the focus squarely on the diagnostic capabilities of German laboratories and the agility of their infectious disease surveillance networks. As the situation develops, the primary objective for health officials is to confirm the presence of the virus through molecular testing and to determine if this instance represents an isolated spillover event or a signal of a broader shift in the local viral landscape. The clinical response centred in Düsseldorf involves a coordinated effort between local hospital staff and national reference centres for retroviruses and emerging infections. When a patient is placed in a bio-isolation unit, under these circumstances, it triggers a rigid set of protocols designed to prevent any potential secondary transmission, regardless of whether the virus is currently known to spread between humans. In the case of hantavirus, human-to-human transmission is extremely rare, with the exception of the Andes virus found in South America, however. The precautionary principle dictates that suspected new strains be handled with the highest level of biocontainment until their biological properties are fully understood. Laboratory technicians are likely utilizing real-time polymerase chain reaction, or PCR, assays to detect viral RNA in the patient's blood or tissue samples. If these initial tests fail to match known genomic sequences for common European hantaviruses like Pumala or Dobrava, scientists will move toward next-generation sequencing. This process allows researchers to map the entire genetic code of the pathogen, comparing it to global databases to identify mutations or entirely new lineages. This analytical phase is critical because it moves the investigation from a state of suspicion to one of verified data. At this stage, there are no confirmed reports of disruptions to trade, travel, or shipping in the North Rhine-Westphalia region, as hantavirus outbreaks are typically managed through localized environmental controls rather than broad lockdowns. Public health agencies are likely conducting contact tracing and environmental sampling in the areas recently frequented by the patient to identify the potential rodent reservoir. The contrast between the initial sensational reports and the methodical reality of laboratory science is stark. While headlines may emphasize the shock of the isolation, the medical community is focused on the data points provided by serology and viral load counts. Until the Robert Koch Institute or local health departments release a formal characterization of the virus, the status of the pathogen remains classified as a suspected variant. This period of uncertainty is a standard part of epidemiological investigations, where the speed of information must be balanced against the absolute necessity of diagnostic accuracy. Prevention and ongoing surveillance remain the most effective tools in managing hantavirus risks, both for known strains and suspected new variants like the one reported in Düsseldorf. Public health guidance generally emphasises the importance of rodent control and the safe cleaning of areas where mice or voles may have nested. Because the virus is often transmitted through the inhalation of dust contaminated with rodent excreta, officials recommend that individuals in affected areas avoid stirring up dust in confined spaces like sheds, barns, or attics. Using a disinfectant to dampen surfaces before cleaning and wearing a high-efficiency particulate air mask are standard preventative measures. The current situation in Germany serves as a reminder of the limits of our knowledge regarding viral evolution. As human habitats expand and climate patterns shift, the interaction between humans and rodent populations can change, leading to the discovery of previously unrecognized pathogens. Surveillance systems are specifically designed to catch these anomalies, and the rapid isolation of the patient in Düsseldorf suggests that the early warning mechanisms within the German healthcare system are functioning as intended. There remains much that is unknown about this specific case, including the exact source of the infection and the eventual clinical outcome for the patient. However, the move to bio-isolation provides a controlled environment where these questions can be answered without risking public safety. Honest reporting dictates that we acknowledge the gap between a suspected case and a confirmed public health threat. As laboratory results are finalised, the focus will shift from emergency response to long-term monitoring of rodent populations in the region to see if a new strain is indeed circulating in the wild. For now, the situation is being managed by specialised teams trained in high-consequence infectious diseases, and further updates will depend on the speed of the genomic sequencing currently underway. Staying informed through official health department bulletins is the best way for the public to navigate these developments without falling prey to undue alarm. Subscribe for more updates on global health surveillance and emerging viral reports. The clinical response currently unfolding in Düsseldorf is centred at the University Hospital's specialised bio-isolation unit, a facility specifically engineered to manage pathogens that require the highest levels of biocontainment. In Germany, such units are part of a coordinated national network designed to isolate high-consequence infectious diseases from the general patient population while providing intensive care. The decision to trigger these protocols for a suspected hantavirus case indicates that medical authorities are treating the potential for a novel strain with extreme caution. Under standard operating procedures, once a patient presents with symptoms consistent with hemorrhagic fever with renal syndrome, such as high fever, acute back pain, and signs of kidney dysfunction, and a history suggesting exposure to rodents, the case is immediately escalated to regional and national health departments. The involvement of the Robert Koch Institute, Germany's central federal institution for disease control and prevention, is standard for any pathogen that appears to deviate from known endemic patterns. While the initial reporting emphasises the shock of the isolation, for the medical teams involved, this is a highly regulated and practiced sequence of events. The primary objective is to move beyond the clinical suspicion and achieve a definitive molecular diagnosis. This begins with the collection of blood and tissue samples, which are transported under strict biosafety protocols to the National Reference Centre for Hantaviruses. The laboratory investigation is a multi-stage process. Initially, technicians utilise real-time polymerase chain reaction, or PCR, to look for the genetic signatures of common European hantaviruses, such as the Pumala virus, which is typically carried by bank voles, or the Dobrava Belgrade virus, associated with wood mice. If these tests return negative results despite the patient's clinical presentation, the investigation shifts to metagenomic next-generation sequencing. This advanced technology allows scientists to sequence all the genetic material in a sample, effectively mapping the entire genome of the pathogen without needing to know exactly what they are looking for beforehand. This is the only way to confirm if a strain is truly novel or if it is a known virus that has undergone significant mutation. During this diagnostic window, the patient remains in a strictly controlled environment. Bio-isolation units utilize negative atmospheric pressure to ensure that air flows into the patient's room, but cannot escape without first passing through high-efficiency particulate air, or HEPA, filters. Staff entering these zones must wear specialized personal protective equipment, often including powered air purifying respirators. These measures are taken not because hantaviruses are known for efficient human-to-human transmission, which is historically very rare, but as a safeguard against the unknown biological properties of a suspected new variant simultaneously. An epidemiological investigation is likely underway in the patient's local environment. Public health officials work to identify the specific geographic location where the spillover event occurred. This often involves environmental sampling and the trapping of local rodent populations to determine the prevalence of the virus in the wild. By comparing the viral sequences found in the patient with those found in local rodents, researchers can confirm the reservoir species and the exact source of the infection. This data is critical for determining if the Düsseldorf case represents a localized incident or the emergence of a broader public health challenge. While media reports focus on the immediate drama of the isolation unit, the scientific reality is one of meticulous data collection and genomic analysis. There have been no confirmed reports from health agencies regarding any wider restrictions on travel or commerce in the Düsseldorf area, as hantavirus management typically relies on targeted rodent control and public awareness rather than broad quarantine measures. The contrast between the sensationalized headlines of a city in shock and the methodical, evidence-based approach of the German infectious disease framework highlights the gap between public perception and clinical reality. Until the laboratory results are verified and published by the relevant authorities, the status of the pathogen remains a clinical suspicion, with all medical actions dictated by the precautionary principle to ensure maximum safety for both the patient and the public. In the wake of the admission of a patient to a bio-isolation unit in Düsseldorf, public health guidance remains centered on established protocols for the prevention of zoonotic transmission. While the investigation into a suspected new strain of hantavirus continues, the core methods for reducing risk are well documented and rely on breaking the link between rodent reservoirs and human environments. Hantaviruses are primarily shed in the urine, feces, and saliva of infected rodents, such as the bank vole or the yellow-necked field mouse. The most common route of infection is the inhalation of aerosolized viral particles, which occurs when dried rodent waste is disturbed during cleaning or construction activities in confined spaces like sheds, garages, or attics. To mitigate this risk, health officials consistently advise against dry sweeping or vacuuming in areas where rodent activity is evident. Instead, the recommended practical takeaway is to thoroughly soak affected surfaces with a disinfectant or a 10% bleach solution before cleaning. This wetting process prevents viral particles from becoming airborne and significantly lowers the chance of inhalation. For individuals working in high-risk environments, the use of high-efficiency particulate air masks and gloves provides an additional layer of protection. Furthermore, sealing entry points into homes, specifically any gaps larger than 1/4 of an inch, and removing outdoor food sources for rodents are critical long-term prevention strategies that remain effective regardless of the specific viral strain involved. The broader surveillance landscape in Germany is characterized by a sophisticated monitoring system that tracks ecological shifts contributing to viral outbreaks. Public health agencies, including the Robert Koch Institute, monitor what are known as mast years, periods of heavy seed production by beech and oak trees, which lead to explosions in the rodent population and a subsequent rise in human hantavirus cases. This environmental data is synthesized with clinical reports to create an early warning system for healthcare providers. In the case of the Düsseldorf patient, the swift transition to bio-isolation reflects the agility of this surveillance network. However, there are significant limits to current knowledge that the public must acknowledge. Until genomic sequencing is completed, it is impossible to determine if this suspected new variant possesses a higher mortality rate, increased transmissibility, or resistance to standard supportive therapies. The term suspected new variant is a technical classification used during the diagnostic phase and does not inherently mean a pandemic-level threat. It is a signal for increased clinical vigilance rather than public alarm. Ethical reporting in these circumstances requires a balance between transparency and the prevention of misinformation until official lab results are verified. Any claims regarding the origin or virulence of the pathogen remain speculative. The ethical handling of this situation also involves protecting the privacy of the patient while fulfilling the public's right to be informed about potential health risks. Public health officials are tasked with communicating the level of threat without resorting to sensationalism that can lead to social stigma or unnecessary economic disruption. The use of specialised bio-isolation units is a sign of a high-functioning healthcare system operating under the precautionary principle, ensuring that even the most remote risks are managed with maximum rigour. As the medical community waits for data from the National Reference Centre for Hantaviruses, the focus shifts to a calm and methodical assessment of the local viral landscape. This incident underscores the importance of the One Health approach, which recognises that human health is inextricably linked to the health of animals and the environment. By monitoring rodent populations and maintaining high standards of clinical isolation, authorities aim to contain the pathogen at the source. The situation in Düsseldorf serves as a critical case study in modern infectious disease management, highlighting the intersection of advanced laboratory science and traditional public health fieldwork. As more information becomes available through verified official channels, the guidance for the public will be updated to reflect the specific characteristics of the virus in question. For now, following